Hello and welcome to Get Yourself Optimized. Today's guest is Dr. Aubrey de Grey. He's a biomedical gerontologist based in Mountain View, California, and is the Chief Science Officer of SENS Research Foundation, a California-based 501c3 biomedical research charity that performs and funds laboratory research dedicated to combating the aging process. He truly is an expert on aging and anti-aging, or reversing the disease of aging. Aubrey, it's so great to have you on the show.
Well, thank you for having me.
So first of all, what the heck is senescence? Because that's part of your brand, the SENS Research Foundation. The first S is, or is it the second S, is for senescence, right?
The name of the foundation, SENS Research Foundation, it stands for Strategies for Engineered Negligible Senescence, but that's a hell of a mouthful, so we don't... So the reason that I gave it that name, and indeed that I gave the concept the name SENS 20 years ago, was that it's the word that people in the field of the biology of aging have been using for a long time to try to be more precise about what they mean by aging. Really, though, I think that's a little bit of a waste of time. I think that the word aging is properly understood anyway by everybody to mean the undesirable aspects of people's lives that occur as a result of physiological decline late in life. But that's all that senescence means, the bad parts of aging, to distinguish it from the things like becoming more knowledgeable.
Got it. And there's something in our bodies that happens as part of our normal upkeep at the cellular level. It's programmed cell death. And one thing that oftentimes gets us in old age is cancer. And how does these two things interplay, the programmed cell death and cancer?
In a complicated way. So programmed cell death, as you say, is programmed. And that means that it's a good thing. It is a way in which the body gets rid of cells that it doesn't want. This happens during our early life, even before we're born, because the growth and the shape and the structure of many of our tissues is created by cells actually going away in certain cases in order to make the right shape. It's also an important process throughout life in the elimination of cells that have become damaged in a kind of irretrievable way, so that it's just better to get rid of them and replace them with a division of another cell rather than trying to repair the cell. And actually, a significant part of aging consists of the body's failure to do that, because sometimes cells get into a state which is not only aberrant in that they're doing more harm than good, but also it is in one or another way resistant to the process of programmed cell death. And indeed, not just programmed cell death, but also the cell being killed by other cells of the immune system, which is another way that the body has for achieving this goal. Now, the relationship with cancer comes there, because cancer, of course, more or less by definition, is the excessive division of cells dividing when they're not supposed to. And certainly, one way to make sure that that doesn't happen, or at least that it doesn't happen to an extent that kills us, is to kill those cells, because that is one type of being aberrant. And certainly the immune system is extremely good at that. And indeed, cells that have become precancerous very often actually die on their own, even without the help of the immune system, so as to get themselves out of the way. So when we get a cancer that's big enough to kill us, an awful lot of things have to have gone wrong simultaneously. And that's a good thing, because it's the only reason why cancer doesn't happen earlier in life.
Mm-hmm. So what are some of these things that protect us from cancer cells that are probably at this very moment in our bodies, right? Statistically speaking, wouldn't you imagine that we all have cancer cells in our body that are being destroyed by our immune system?
Oh, for sure we do. There's no question about that. In fact, even in childhood, we would have some. Because mutations that are the underlying cause of cancer are a reasonably common event. In fact, the cell undergoes massive, millions and millions, each cell in our body, millions and millions of aspects of damage to its DNA every day. And of course, almost all of those lesions are repaired properly by a very sophisticated array of systems in the cell, but some of them are not, and those mutations gradually accumulate. Now, most mutations don't matter very much. Either they just kill the cell, or they just stop the cell from doing something that it should be doing, or they don't do any harm at all. but occasionally they make the cell more inclined to divide than it ought to be. And then we've got all these other things, some of which I just mentioned, that the cell has to do, which are caused by other mutations that allow it to become a bona fide cancer. So it has to figure out how to avoid the immune system. It has to figure out how not to listen to its own signals saying something's wrong, please die. It needs to avoid other processes that get it into a state called replicative senescence, where basically it has a very severe break on the ability to divide. It's got to do things to break down the proteins or the extracellular matrix that are around the cell that would otherwise just physically constrain it from dividing and growing. The list goes on and on. In fact, many years ago, some important cancer researchers wrote a paper on this called The Hallmarks of Cancer, which essentially tried to spell this out and summarize the state of knowledge with regard to how clever cancers need to be and how we might go about making us more resistant to cancer or getting rid of cancers. And that term, hallmarks, has actually been reused recently to describe the situation with aging. About 5.5 years ago, a group of gerontologists in Europe put out a paper which, to be honest, was pretty much a restatement of something that I'd written at the beginning of SENS about 11 years previously. But they called it the hallmarks of aging, just kind of by reference to this cancer thing. And I'm very happy about it because it got a lot of attention. People understood it better than they had understood it when I said it a decade earlier. And it's cited all the time. It's considered completely mainstream now.
Got it. And these hallmarks of aging, how many of them are there? And were there any new ones that they came up with besides the ones that you wrote about 11 years earlier?
Yeah, great question. So actually, no, the opposite happened. They left some of mine out, but the number that they got to was larger than my number. They talked about 9, where I only talked about 7. The discrepancy there is simply because they split it up in a different way. The way that they split it up didn't really have any reference to the type of interventions. The interventions that they were looking at kind of overlapped in a different way, whereas the ones that I talked about were divided very much on the basis of lining up one-to-one with types of intervention.
So what are these hallmarks, the 7 that you've come up with, and then what are the interventions for these?
Okay, this is going to be a 5-minute answer.
That's totally fine.
The first one is loss of cells dying and not being replaced by cell division. So, of course, if that happens in a given tissue, a given organ, then the number of cells progressively goes down. Eventually, there are not enough cells to do the job that the tissue needs to do, and we have a problem. A great example of that in aging is Parkinson's disease, which is caused by the loss of a particular type of neuron in a particular part of the brain. And the fix is, of course, stem cell therapy. We put cells into the body that have been programmed in the lab into the right state so that they know what to do to divide and transform themselves into replacements for the cells that the body is not replacing on its own. So that's the easiest one of the 7. It's also the one that's furthest along, not surprisingly, and it's certainly the one that people know about the most. Next one is cells dividing when they're not supposed to. We just dealt with that. That's cancer. And of course, there are many different approaches to cancer. The one that I'm most excited about right now is cancer immunotherapy. Of course, everyone's excited about that over the past few years because of the great progress that's been made, largely arising from 2 huge breakthroughs that were made nearly a decade ago now to make the immune system better at getting rid of cancer cells. But you have seen nothing yet. There's plenty more in the works. Okay, so tease us a little more with what's coming. Like, what do you envision and when? Yeah, what's coming is really enhancements of what we can do already that resulting from improving technology at the level of sequencing and at the level of manipulation of DNA and proteins for that matter. So, for example, one thing that cancers do, all cancers, is they produce proteins that are not produced by normal cells. And this happens simply because of random chance. As I mentioned earlier, a bunch of mutations needed in a cancer cell to get it to be cancerous, but you only get those as a result of having lots and lots of mutations that are just bystanders, that don't have anything to do with the cell being cancerous, but they still have a signature in the sense that they cause these unusual proteins to be made. And those unusual proteins, of course, presented on the surface of the cell, just in the same way that every other cell does, a signal to the immune system to see the identity of the cell. And if those different proteins are seen by the immune system, then they're seen as foreign, and the immune system has a chance of killing the cell. Now, it's a bit of an arms race because the cancer will also develop ways to resist the immune system, as I mentioned. But the more the immune system is tuned to eliminate cells with these what are called neoantigens on them, the more the balance of that arms race is tipped in favor of the immune system and against the cancer. So one very cool thing that people are trying to do is to sequence the entire genome of cancers and figure out what neoantigens they're going to make as a result of the sequence, and then make large numbers of antibodies, different antibodies to different neoantigens, so that the immune system can attack all of them simultaneously more aggressively than it normally would. And this seems to be working very well. So that's just one example.
Oh, very cool. And gene editing inside of our bodies, that's made possible by a technology that I recently heard about called CRISPR in the last couple of years. Is that something that would play a part in what you're describing with the cancer immunotherapy?
Yes and no. So the thing about most of the ways in which we would do cancer immunotherapy is that it involves taking cells out of the body and manipulating them genetically in the lab and then putting them back. And CRISPR is certainly very useful for doing that, but it's not the only technology that's useful. In general, what you can do to the DNA of cells in a culture dish is much more versatile. There's much more options than what you have in the body itself, because you've got, of course, much more control over safety.
Okay, so that's the second of the 7 is the cancer, and then cancer immunotherapy as a mechanism to deal with. What's next?
So there is then another way in which you can have too many cells, bad cells, and that is if cells are not dying when they are supposed to. So I mentioned that that's one of the things that cancers need to do, is to resist their own cell death machinery. But cells that are not necessarily dividing too much also have this problem, potentially. And indeed, some of them do. We see these cells. They're normally called senescent cells, but actually I like to refer to a somewhat broader category, because the term senescence has a technical meaning that is a little too narrow. Senescent cells, or death-resistant cells, get into this state. They're doing more harm than good, but they're not dying. And so, again, we want to try to make that happen. Now, there are a lot of similarities with the cancer situation. In fact, some people have described senescent cells as cancer cells that are not dividing, which is a little bit, weird. But the intention is to indicate that similar kinds of things can be done with the immune system to help us to get rid of these senescent cells. However, because the cells are not dividing, simpler methods, like simple drugs, have a much better chance of success than they do for cancers. Cancers have the problem, of course, that they have natural selection on their side, with constant mutations happening to allow the cancer to resist whatever therapy we throw at it. Senescent cells don't have that. So senescent cells are actually relatively easy to get rid of, and that's why we are now in the very wonderful position of having really a lot of companies, like half a dozen companies, following a disparate array of approaches doing this, whether it's with small molecules, with genes, or with immune enhancement or whatever. And some of these companies are getting into clinical trials already. They're getting very well funded by visionary angel investors and even by more established investors. So life is good around the senescent cell world.
Okay.
So the rest, the other 4, are not to do with cell number. They are to do with molecular changes. And 2 of those things are inside cells, and the other 2 are outside, in the spaces between cells. So first of all, the ones that are inside cells, we've got mutations in the mitochondrial DNA. So we've talked a bit about mutations in our regular chromosomes, which lead to cancer. And I have argued for a long time that actually mutations in our regular chromosomes that don't lead to cancer basically don't matter, that the evolution has been forced to create a system of DNA repair and maintenance that's so good in order to stop us from getting cancer before we've had offspring, right, that it's unnecessarily good for everything else. So yes, it's still a controversial hypothesis, really, whether the mutations in the nuclear genome that do not lead to cancer actually matter enough. In other words, whether they accumulate during a normal lifetime to a level that's actually bad for us. I still believe what I wrote more than a decade ago, 15 years ago, that no, actually we can ignore those because cancer is the only problem. But that doesn't tell us about the mitochondrial genome. The mitochondrion, of course, is this very important part of the cell that does the chemistry of breathing. In other words, it combines oxygen with nutrients in a way that extracts energy from those nutrients that the cell uses for everything else. And the mitochondrion is the only part of the cell that has its own DNA. Now, it turns out that that's a really bad place for DNA to be, because it's right next to this chemistry of breathing. And that chemistry of breathing creates these toxic molecules called free radicals which damage DNA, among other things. So sure enough, the mitochondrial DNA accumulates mutations far, far faster than the nuclear genome. In fact, there are other reasons why that happens, not only the ones I've just mentioned. The result is, those mutations, most people agree, they actually do matter in a normal lifetime. So we better do something about that. And unfortunately, fixing those mutations is really tricky, really, really hard. But we have another option, which was first suggested more than 30 years ago by a group in Australia, and which was pretty much universally abandoned as being too difficult. But over the past 10 or 15 years, at SENS shown that that was premature, and we're pretty close to making it work. And the approach is to put backup copies of the mitochondrial DNA into the nuclear genome, into the regular chromosomes, where they are not subjected to such a high threat of mutation. Now, if you put DNA in the wrong place, it's not going to work. So something has to be done to compensate, to tweak the DNA so to make the proteins that it encodes go back into the mitochondrion and do their job, even though the DNA is in the wrong place. But luckily, that's not as hard as it sounds, because the mitochondrion has a lot of proteins, more than 1,000, and the mitochondrial DNA only encodes 13 of them. The 13 in question are very important, vital ones, but that's all there are. So that we've already got machinery naturally in the cell that does this importing into the mitochondrion of thousands, more than 1,000 anyway, proteins that are naturally encoded in the nucleus. And therefore, the only requirement is to co-opt that same machinery for these additional 13 proteins. And that's basically what we've been able to do. Okay, so the next one, number 5, is also inside the cell. And this is a much simpler one to describe. It's just garbage, waste products that accumulate inside the cell. The cell makes waste products of all manner of kinds all the time as byproducts of the processes it needs to perform to keep us alive. And almost all of those products are destroyed. Either they're destroyed in the cell or they're eliminated by excretion. Just they're chucked out of the cell and then out of the body. And that's fine. But there is a small minority of byproducts which are not. This is simply because they are very rare byproducts, and therefore they accumulate very slowly, so slowly that they don't cause any problems for the cell until late life. And of course, late life is the time when evolution no longer cares about us because we have already reproduced. So those things are not destroyed, and they're also not excreted. They accumulate inside the cell, usually in a kind of garbage disposal machinery called the lysosome. And eventually they do get to be problematic. And this is why we get things like atherosclerosis from the accumulation of oxidized cholesterol, or indeed macular degeneration, which is caused by the accumulation of a derivative of vitamin A. So we've got to fix this. And of course, the right fix is simply to clean out that garbage. Turns out that the very effective way to do that is to identify bacteria in the soil, typically, that already have genes, enzymes, that are able to break down this garbage. Now, rather in the same way that putting a gene that's normally in the mitochondrion into the nucleus, if you take a gene that's normally in bacteria and you put it into a human cell, it doesn't work. But again, there are tweaks we can do that might make it work. These tweaks are not necessarily obvious, but long story short is that in both the cases I just mentioned, atherosclerosis and macular degeneration, our group was successful in achieving this and thereby augmenting the garbage disposal machinery of the relevant cells. And both of those projects and research themes have now moved forward far enough to be spun out into companies that are indeed going into clinical trials within the next year or two. Next one along is we're now outside the cell. And again, the actual nature of the problem is garbage, waste products. You may think it's a little strange that I would want to list them separately from inside the cell, but the reason why is because they, again, our approach to getting rid of them is very different. In this case, it turns out that all we need to do is get the stuff inside the cell. In other words, get cells to engulf this stuff. And that's because the machinery that we naturally have outside the cell for destroying garbage is really primitive as compared to what we have inside the cell. That means that stuff accumulates outside the cell, even if it's actually relatively easy to destroy. And if it were only inside the cell, it would already be toast. So that's what we make happen. This was actually pioneered in the case of Alzheimer's disease more than 20 years ago. And actually, that was one of the inspirations for the whole SENS idea that I first put forward in the year 2000. It was shown 20 years ago that one could eliminate the amyloid plaques in Alzheimer's disease in a mouse model It took a long time to get from there all the way to making the thing really work in humans, but it definitely works now. There are many different, I think we're now up to 4 different vaccines that big pharmaceutical companies have created that definitively, all the way through clinical trials, they really do remove this amyloid. They don't have all that much of an effect on cognitive function in Alzheimer's patients, but that's no surprise because there's plenty of other things that also go wrong in Alzheimer's, so this is just the first step towards a cure for Alzheimer's. However, we've taken this same thing forward in relation to other types of extracellular garbage, and we've been quite successful. There's one type that happens in the heart, which is apparently very prominent in terms of the cause of death of extremely old people, people over the age of 100. And it may be important at earlier ages as well. That hasn't been studied enough yet. However, we've developed an immunotherapy to get rid of that, again, to cause cells to engulf it, or in this case, actually just to chop up the stuff without getting it inside the cell. And that seems to be very effective. So that, again, has also been spun out into a separate company now that's receiving funding from elsewhere. All right, so I'm now down to my last one, which is, again, outside the cell. But in this case, it's not to do with garbage. It's to do with chemical modifications of stuff that we do not want to destroy. And the stuff that we don't want to destroy is called the extracellular matrix. It's a kind of lattice of proteins that gives tissues their physical properties, like elasticity. And those physical properties are very important because, for example, in the major arteries, we need elasticity so as to buffer the heartbeat, so that the fragile, more distal parts of the circulatory system, the capillaries, do not get damaged. And sure enough, what happens over time is that that elasticity diminishes. The major arteries become stiffer, and the result is the heart has to beat harder in order to get blood around. So we have an increase in blood pressure, which causes all the problems we're familiar with. So we need to fix this. We need to restore that elasticity. And it turns out that the molecular basis for the loss of elasticity has been well understood for a long time, and basically it's the result of spontaneous chemical reactions between the extracellular matrix and sugar in the bloodstream. Those reactions are understood, and the chemical structures that result from them are also identified, or at least we believe that the main ones have been identified, and they're very different from the structures that the body creates on purpose. So, of course, that means that in principle it should be possible to generate drugs or enzymes that are nicely selective, that will break these unwanted accumulating chemical crosslinks without having side effects. And again, we've had great success in that area, work that we funded at Yale University, and that too has now been spun out into a company that's getting very good funding.
Wow. It's amazing. And your research efforts are really making a difference in moving some of these interventions forward and then companies getting founded. Are you a stakeholder, a shareholder in these different companies that are being formed, or how does that work?
Right. We're very, very proud of our successes here. I think, we can now call it really our business model is to pursue the most challenging aspects of damage repair for aging for as long as it takes to get to a sufficient level of proof of concept that investors can see a value proposition. We do take a stake in these companies when we spin them out, but only a small one, because we feel that it's important not to dilute other investors. And also, let's face it, these are early stage companies. They won't have products and so on for quite a long time. So we don't really see it as a meaningful near-term source of revenue for the foundation. But in terms of the influence that we have, it's much larger than that. In addition to our spin-outs, there are dozens of other companies doing very aligned work in one way or another, doing damage repair. And, of course, we want those companies to succeed as well. So I am advising such people all the time. And, of course, investors, likewise, understand that I and the foundation, at the nexus of all of this, we're the go-to people for information, so they're all coming to me as well. And I'm spending a lot of my time these days just making introductions, just making things happen, making sure that money goes to the places where it's going to be most effective.
Interesting. Okay. Now, back to these 7 different aspects of aging and the interventions. With cancer, with number 2, you mentioned cancer immunotherapy as a very promising intervention. Of course, you didn't mention chemotherapy or radiation or surgery as interventions that are promising. What are your thoughts on these different kind of old school models of dealing with cancer?
Well, I mean, of course, we mustn't neglect them. The fact is that as of today, Cancer immunotherapy does not work effectively on all cancers by any means. Probably going to be a little while before it does. And other more radical approaches to eliminating cancer, like, for example, inhibiting the ability of cancer cells to extend the ends of their chromosomes, which is an approach that we've pursued quite aggressively over the years. That again is some way off. So we have to, work with what we have. My friends who are the MDs rather than the PhDs, of course, that we're working with what we have. However, everybody knows that the goal is to develop therapies that are less invasive and much more free of side effects. Everyone knows that any intervention that is purely focused on selectively eliminating cells that are dividing rapidly is going to have big side effects, because we've got plenty of other types of cell that divide rapidly. And indeed, some cancer cells don't divide as rapidly as all that. And those are probably the cells that it's most important to eliminate.
Got it. Now, one of the things that you mentioned going through the 7 different aspects was garbage inside the cell and outside the cell, and then the garbage collection that needs to happen as part of our normal living, respiration and all that. Where does the heavy metal toxicity fit in? Because we get exposure to mercury from eating fish and lead, and there's also PCBs that are still in the environment. There's glyphosate. There's even 5G and just EMF kind of exposure that is causing havoc to our cells. Where do these different things fit into that kind of garbage collection model inside and out of the cell?
First, let's talk about the radiation aspect that you mentioned. Of course, radiation itself does not accumulate. What you're saying is that radiation might have effects on the rate of accumulation of other types of damage. And to be honest, I think that the evidence for that is extremely limited. I don't think the case has really been made. But the key point is, even if it is the case that radiation from external sources is accelerating the accumulation of different types of damage, then so what? Because if those types of damage are already occurring at a slower rate and we're repairing them by the corresponding methods I've just mentioned, then all that says is that we would need to repair them a little bit more quickly, a little bit more thoroughly, and that's no problem. With regard to actual substances coming in from outside, like heavy metals or whatever, you're quite right, that's an important thing to consider. But again, the evidence that it contributes significantly to most people's health at late ages is pretty much absent. I don't think we can really make that case. Of course, one can have extreme situations of exposure, but I'm talking about most people. Nevertheless, that doesn't necessarily mean we should ignore it. So I think that it is important to recognize that there is a way out of this, which is oscillation in how much fat we have. Fat cells, adipose tissue, tends to accumulate heavy metals and such like preferentially, which means that if we want to get rid of it, then one thing we can do is simply starve for a bit, because that means that our fat cells will essentially metabolize their contents, and what happens is, sure enough, they release their contents, including these things like heavy metals, into the circulation. Now, of course, we don't want to do that too rapidly, because then the metals will just go elsewhere, but a small amount of release will simply cause an excretion of the relevant substance through the kidneys, and therefore the overall amount of heavy metal that remains in the body will have been reduced. And of course, then we can start eating again and get back to where we were, except that we've lost those heavy metals. So that's the nice low-tech approach to that particular problem.
Yes, and that's intermittent fasting that you're referring to?
Well, of course, intermittent fasting has connotations. That is a method that people have been promoting for much more general good health, especially during late life. And it's essentially just a refinement of the general concept of calorie restriction, of eating less than one would really like. And, it has some benefits, but it certainly does not at this point seem to be likely to significantly extend life.
Yeah, okay. So what are you doing to, like you personally, to extend your life, to increase your health, like supplements and therapies and various technologies? What are you up to?
Yeah, I don't do anything, but
Okay.
I really like to say, do as I say rather than doing as I do, because there are two reasons why I don't do anything. The first one is I'm just really lucky. I have had the good fortune to be tested I think 5 or 6 times now over the past 17 years or thereabouts. Really high-end, thorough tests of my physiological age, measuring 150 different things in my blood and so on. I always come up way younger than my actual age. So, I'm in a position where if it ain't broke, don't fix it, really. So even, I have to be pretty conservative. I just seem to be the kind of person who can eat and drink exactly what they like and, nothing really happens. I don't even need to do much exercise. But also there's a second thing, which is I want to save lives and I work very hard to do so. And the result is I don't get nearly enough sleep. And that's definitely bad for me. I figure, it's only slightly shortening my life and I may be lengthening my life by my own work. So, maybe it's a net win. But yeah, don't do as I do.
Got it. Okay. And what are those tests that you do that check all these 100 or whatever different attributes? Is that like the health nucleus test from Human Longevity Inc. or something different?
Yeah, no, it's older than that. The tests that I've had done do not involve genetic tests or epigenetic sequencing or anything like that. And in due course, I definitely will move up to that kind of thing. But the ones I've had done just measure the obvious things like insulin and, triglycerides and oxidized lipids and so on. And, everything else, all the kinds of things that typically change with life, various hormones. And I always come out on the young side. But also cognitive tests, physiological tests, how long I can go on a treadmill, stuff like that.
Got it. Okay. Now, you mentioned epigenetics. And for our listeners who are not familiar with that concept, could you just describe the difference between genetics and epigenetics and why it's important?
Yeah, sure. So everybody knows that genes consist of this molecule called DNA, which is a sequence of 4 different components, very, very long sequence, 3 billion of these things in 2 copies in each cell. And that defines what proteins our cells can make. But it does not define on its own what proteins our cell does make. Because, of course, different cells have different roles in the body, and in order to perform those different roles, they have to make different proteins in different quantities. Now that, of course, has to be somehow implemented in the cell. The cell has to kind of know what it's creating and what it's not creating, and that is done by what's called the epigenetic state of the cell, which is at the molecular level achieved by certain chemical modifications of these components of the DNA, things like methylation, and also by chemical modifications of proteins that are in the proximity of the DNA. So, for example, there's a type of protein called a histone, which the DNA is normally wrapped around. And there are certain modifications to histones that determine whether a particular gene in that vicinity of the DNA is going to be turned into a protein or not. So that's really the epigenetic state of the cell. And the epigenetic state does not only differ from one cell to another depending on what the cell's role is, it also changes throughout life. And recently, over the past few years, there's been a great deal of focus on this because people have been able to come up with really rather good indicators of the biological age of somebody by looking at what's called their epigenome, specifically at this methylation thing I just mentioned. Of course, what they have shown is, first of all, that the thing measures your chronological age, which is not useful in itself because we already have birth certificates. But then they can look at deviations from that prediction, and they can ask whether somebody is looking epigenetically older or younger than their chronological age. And that's where the research focus is these days. There's a huge amount of great stuff happening in that area.
And is there a particular test or analysis that you would recommend to check your epigenetic age, your biological age versus your chronological age?
Oh, no, certainly not. But firstly, because I'm not dumb enough to endorse things and by default unendorse other things, but also because things are just moving too fast. The quality of these tests is improving very rapidly right now, and there's more to it than what I've said so far. It's not just the epigenetic tests. There's also tests of the same kind looking at what's called the proteome, which is the set of proteins that exist in, for example, your saliva or your blood. There's other work being done to combine these things, to look at, for example, also metabolites at the same time, even metabolites whose function we don't know, but which nevertheless seem to change with age, and therefore they could be useful indicators. So there's a great deal going on.
Got it. Okay. And there's also something that happens as we age where the caps on our chromosomes, the telomeres, get shorter and shorter. So there's some implications there as far as aging, correct?
Maybe, maybe not. So way back, more than 50 years ago, in fact, nearly 60 years ago now, it was indeed discovered that when a cell divides, the ends of the chromosomes get shorter. Actually, the first thing that was discovered 60 years ago was that cells simply get into this state called replicative senescence, where they don't divide anymore. And it was about 10 years later that people figured out what was really going on, about why these cells stopped dividing, which was, as you say. Now, the reason for this is very fundamental. The process of DNA replication has only evolved once in the whole history of life, and the way that it has evolved just turns out not to be able to replicate the ends of DNA. When DNA replication first evolved, we didn't have that problem because bacteria, which of course came first, don't have linear chromosomes. Their DNA is in a circle. But we do have that problem. And so in order to get around it, evolution has created this special enzyme called telomerase, which adds DNA back on the end of the chromosome. You only have to add a little bit because only a little bit of DNA is lost when a cell divides. It still adds up unless you have this compensation. So what does all this mean? Well, the first thing it means is that if you have a lot of cell division in the body, then the cells are just going to eventually die, or at least stop being able to do their job. But if you look at what the body is actually made of, there are not many cells that are doing that. Most of our cells don't divide at all, or they only divide very occasionally on demand. Like, for example, cells in the lower layer of the skin, called fibroblasts, they divide pretty much only when there's a wound. They divide like crazy then, they're really good at it, to close the wound, and that's actually why they are the cell of choice for doing these experiments in which the concept of the phenomenon of replicative senescence was first discovered. But they don't divide much normally. So actually, when you're doing an experiment with fibroblasts showing this phenomenon, you're not looking at something that's representative of what goes on in the body. You're making these cells, essentially you're tricking them into thinking that they're in an infinite-sized wound that they can never quite close, so they're dividing far more than they would ever need to, even in a whole lifetime, normally. Now, there are some cells, however, that do divide regularly, so frequently that they would get into this problem. For example, the stem cells of the upper layer of the skin, the epidermis, that's an example. Also the stem cells of the blood. But the thing is, those cells use this compensation mechanism. They create a little bit of this enzyme called telomerase, so they don't have a problem. So what this all adds up to is that it's not at all clear that replicative senescence actually plays a significant role in aging of the human body. There are some areas where it may, especially some aspects of the immune system. It looks as though there may be something going on, but it's not clear. The flip side is that cancers actually do have a lot of this enzyme telomerase, and that's what allows them to grow and grow and grow. If they didn't have it, then they would actually divide themselves into oblivion way before they got to be big enough to be even noticeable, let alone to kill us. So a perfectly reasonable approach to attacking cancer is to try to stop cells from expressing telomerase, this enzyme. We've had a go at that. It's pretty hard, but it might be an important way to go, especially if cancer immunotherapy encounters obstacles that stop it from being the real McCoy. So that's where we are with the telomere question at the moment. It's still very complicated.
Okay, got it. Now, one thing I've heard from some folks like biohackers in particular, like Dr. Jack Kruse, said that a lot of our aging and our chronic disease is happening because of the mitochondria and chronic mitochondrial diseases. There's a term, I guess, for people who are focused on improving their mitochondrial health. They're called mitochondriacs. And could you say a bit more about that?
Well, actually, the term mitochondriac is a kind of tongue-in-cheek term that people who do research on mitochondria tend to use for themselves. So I already spoke about one aspect of mitochondrial aging, which is the accumulation of mutations. But actually, even in old age, only a very small proportion of our mitochondria have mutant DNA. That small proportion is not evenly distributed, however. You have some cells that are completely taken over by mitochondrial mutations, and that's probably very important to why mitochondrial mutations matter and how they actually contribute to ill health. However, there is a much more general phenomenon where mitochondria simply don't work so hard. They don't make so much energy, extract so much energy late in life. And that's a very curious phenomenon that is still debated in terms of why it happens and how it happens. But it's definitely important. However, what we can say is that it is not autonomous. In other words, it is not a result of some intrinsic thing that's going wrong in the mitochondria themselves, because the mitochondria are constantly being recycled. Even in cells that are not dividing, the mitochondria are dividing every so often, and some of them are destroyed, the other ones divide. There's constant turnover. So there's no way that anything other than the DNA could actually be accumulating damage. Therefore, what's going on is some kind of response to something else that's going wrong in the cell. The mitochondria are not working so well because they are in some way being told not to. Therefore, the next challenge, obviously, is to figure out what the environment of the mitochondrion is that matters. And there's been a lot of focus lately on a molecule called NAD, which is an extremely important metabolite that essentially acts as a reservoir of electrons that is used by mitochondria as how they do their thing. And the amount of NAD in the cell, and also in the bloodstream for that matter, declines with age. And so does the amount of unladen NAD. So in other words, basically the reservoir of electrons is smaller and it is fuller. A higher percentage of the reservoir is occupied. And that seems to be a bad thing. It basically seems to make the mitochondria work less well. So the idea is maybe if we can give supplements that are able to raise the amount of NAD, you don't give NAD itself because it gets destroyed, but you can give precursors of it that are used as building blocks for NAD, then you can increase the size of the reservoir and you can also decrease the fullness of the reservoir by default, so to speak, and that seems to be beneficial. Exactly what this will do for aging overall remains to be seen, but it's a very active research area, both in academia and in the private sector now.
Mm-hmm. Is there anything that you would recommend our listeners do to improve their mitochondrial health while this research is going on?
To be honest, no. I think that at this point, it's too early to say what's going to work well, what's going to not work well. And furthermore, one thing that I always have to point out is anything that I might recommend, or indeed anything that you might read in a book, you should always take with a pinch of salt because it is the lowest common denominator. It's a general statement about what everybody should do. And that makes no biological sense because people differ too much. People vary too much in terms of their metabolic requirements, their metabolic performance. Really, the only general advice that one can give is pay attention to your body. Do what works for you. And, one doesn't sell books by saying that.
Sure. So that means paying attention to what seems to be working and not working when you try different supplements and so forth, but also getting tests that give you real data back on how you're doing too, right? I would imagine that getting regular scans and blood tests and that sort of stuff is going to help you stay on the right path there, right? Because Peter Drucker said that what gets measured gets managed, right?
All of the above, yes. At least we can certainly say that what doesn't get measured does not get managed.
Yeah.
And, of course, not just supplements, but also lifestyle and diet in general and other types of monitoring. A Fitbit is a good thing. All of these things.
Yep. Yeah, I have an Oura ring that I always wear. It tells me how much sleep I got, my activity levels. It'll remind me if I need to get up and move some more. It's very helpful for me and non-intrusive. I also take a bunch of supplements. I take Restore, which helps with communication of my microbiome. I take Cytodetox, that helps with binding the heavy metals and the different toxins. And I take nitric oxide type of lozenge called Neo 40 that helps with nitric oxide production. And just all the regular sort of vitamins and vitamin D and fish oil and that sort of stuff. So I'm curious, and any thoughts on any of those different things I mentioned? Have you had any exposure to any of these different supplements or heard anything that you might want to pass on?
Yeah, no, I really don't have anything to add to what I just said. I mean, one thing I also have to emphasize here is that that's not really my area of expertise anyway. I'm focused on developing regenerative medicine that is currently in its early stages. Other people are a lot more knowledgeable and well-placed than me to advise on diet and supplements and so on.
Got it. Okay. Let's talk a little bit about pets because they're family members for many of us, and they get these chronic diseases and age and their bodies break down. I'm dealing with my cat right now having cancer of the liver and gallbladder, bile duct, sort of thing. And it's beyond the point where it's even to be assisted by surgery. So it's pretty depressing, pretty sad to have to deal with that. I assume that all of these 7 aspects of aging and the interventions are all applicable to pets, to other animals as well. I'm guessing that's where we test, in fact, is with animal models like mouse models and things like that. So any additional thoughts on pets and extending their lives?
Sure, yeah. So, I mean, you're quite right. Of course, this really matters to people. And it has been pointed out for many years that if we had a greater focus on companion animals, whether it's cats or dogs, then we might be able to get better funding for research because people really care. And also because, of course, even though the goal is to actually help these animals, occasionally things are going to go wrong and people get upset when you kill a dog or a cat, but not nearly so upset as they do when you kill a person. So the safety regulatory process is more rapid. Now, of course, cats and dogs are intermediate in size and also in longevity between mice and humans. And that also might indicate that they would be more faithful models. You'd learn more about humans from them than you would from mice. That's not really true, unfortunately. The details of what goes wrong in animals does not really translate in that way. Nevertheless, all that said, there is definitely enthusiasm for doing such work. For example, there's a very important study that's been going on for a few years now at the University of Washington up in Seattle, looking at the effect of rapamycin on dogs. Rapamycin is a rather old drug that has historically been used as an immunomodulator, but over the past several years there's been huge interest in its ability to postpone aging, which was first discovered in mice. It's now believed that it works as a very effective, what's called calorie restriction mimetic. In other words, a drug that makes the body think that it's not getting enough food, that it's in some kind of famine, even when actually you're eating normally. So, yeah, that's a very interesting study, and it's not the only one. There's lots of stuff going on and being planned that's in this space. I'm all in favor of it. The more we learn, the better.
Yeah. So you mentioned rapamycin. What about metformin? I've heard good things about that, and that's a drug that a lot of people suffering from diabetes are taking.
So metformin, as you say, is historically a diabetes drug, another very old drug, and recently, people have got pretty interested in the possibility that it too could be an effective calorie restriction mimetic, or maybe that it could affect aging in other ways. There was a particularly exciting analysis done quite recently which showed that diabetics who receive metformin actually live longer than non-diabetics who don't receive metformin, which is, quite an interesting discovery. Needs to be replicated in other populations, of course, needs to be generalized, but there's certainly plenty of momentum behind the idea that we should explore metformin's general anti-aging properties more thoroughly than has happened so far. And there are plans to conduct a clinical trial of this, even though, of course, metformin being an old drug that's not patentable anymore, no one's going to make any money out of it. And, of course, the result of that is that it's proving to be quite difficult to fund this clinical trial, but I'm hopeful that that will happen in due course.
Back to this idea of funding, you guys are doing so many different research projects and some of these turn into companies. How are you getting most of your funding? Is it from grants? Are you looking for donations from folks like me and my listeners? Like, how can we help, and how does this machinery of technological advances in medicine that you're pioneering, how does that get funded?
Well, that is the right question. Thank you for asking that. We are definitely the engine room of this industry. We pursue projects until such time as they are far enough along to be of interest to people who think they might want to make money in the long run. That's the point at which we spin them out. But the projects that we pursue before that time, to get them to that point, predominantly the ones that other people who do rely on government grants and so on are not doing. And we have to ask, why would they not be doing them? Of course, I'm only talking about ones that need to be done by somebody. It's certainly not because the scientists are not interested. What it is simply that the structure of government funding, and indeed funding from large foundations, is driven by a process called peer review, which is extraordinarily biased, and I would say irretrievably biased, in favor of easy stuff, in favor of low-hanging fruit. The whole academic career structure is driven by getting publications in high-profile journals quickly so that you can get your next grant application funded or your next promotion or whatever. And that is a tragedy. It absolutely catastrophically slows science down because enormous numbers of easy things don't scale. They do not actually contribute to progress towards the most difficult things that are the most important. So we at SENS Research Foundation, we certainly publish, but we don't decide what to work on on the basis of maximizing how often we publish. We publish when we feel like it. This means that we do things that could never be done if they were being funded in a more traditional way. Our work on mitochondrial mutations, for example, we are now recognized as the absolute, far and away, the world leaders in that area because we spent 11 years working on that project before we even got our first publication, and we didn't give up. Now we're way ahead of everybody because they just couldn't do it. They just weren't in a position to work on things that were so difficult. And the only way we do it is by being funded by philanthropy, by making our own decisions independently of vested interests and short-termism. Our budget is still tiny. We still work on an annual budget of only $5 million, which is, if it were bigger, then we would get a lot more done, no question. But we're very proud of what we have achieved with this over the years. Our website, sense.org, has a nice friendly donate button. And if you're in a position to donate more than that, then obviously there's a contact form as well. You can totally get in touch and we'll tell you how to do so. Budget does currently consist of a small number of donations that are large from wealthy people and also a large number of small donations. So every dollar counts.
Okay, great. So listeners, help make a difference in the longevity of the human race and perhaps your loved ones too by donating to the SENS Foundation. And the website for your foundation?
Yep, sense.org. S for sugar, E for elephant, N for November, S for sugar.org.
Awesome. And if somebody wanted to learn more from you, do you have any additional information, articles, courses, webinars, or anything like that you could point them to?
We sure do, yeah. The website, same website, has a wealth of information about what we work on and why and what other people are working on, latest news and so on. It's written for every kind of audience, from complete novices all the way up to professionals. And we think it's very thorough, and I encourage people to go to it. As regards lectures and so on, I spend my life on the road. There is a lot of me on YouTube, so that's one way to find out more. And, of course, we have a conference coming up at the end of this month, actually, in Berlin on March the 28th through 30th. It's called Undoing Aging, and there will be a lot of the most important research being done in this area being showcased. So anyone who can get to that is advised to do so.
Awesome. Well, thank you so much. This was fabulous. I'm just so honored to have you on the show and to share your brilliance. I got to meet you 15 years ago through Foresight Institute. I saw you several times since then, including Neil Strauss's Secret Society, which was really cool as well. And you're doing just amazing things in the world. So thank you and keep it up.
Well, you're very kind. And thank you again for having me on the show.
Yeah, thank you. Now, listeners, get out there and start doing some research and make a difference in the lives of yourselves and your loved ones, and make a donation to SENS because they're doing some really great things, as you've heard. Okay, we'll catch you on the next episode of Get Yourself Optimized. This is your host, Stephan Spencer, signing off.